Welcome everyone, and good afternoon. Thanks for joining us for the second session in the eDNA webinar series. My name is Sam Semino, and I'll be moderating today's webinar. I work for the USGS, where all my time is dedicated to the Pacific Northwest Aquatic Monitoring Partnership, or PNAMP for short. PNAMP, along with StreamNet, co-led the planning of this webinar series, and the planning group included 13 people from a variety of organizations, including David Pilliod, who organized and contacted all of the speakers for this eDNA webinar series. And if you missed it, last week, David Pilliod and Matt Laramie presented the eDNA 101 session. That presentation was recorded and is available online for you to check out. It's on YouTube at the PNAMP channel, and it's also on pnamp.org in the Emerging Technology Information Sessions project page. So for today, here is our approximate agenda. After I wrap up the welcome, we'll do an icebreaker question to make sure everyone is awake after lunch and familiar with using some of the functions of Microsoft Teams. Then we'll hear from our presenter, who's Carl Ostberg. And we are planning about 45-minute presentation with the remaining time allotted for discussion and question and answer. But first, some quick tips on navigating the meeting platform. Please do mute yourself when you're not speaking. You probably already realized that you are muted as you joined, but if you weren't, you can mute yourself on the phone using *6 to mute and unmute yourself. And then if you're using a computer for audio, you mute and unmute using the microphone icon on the toolbar. And with the toolbar, Microsoft has been rolling out changes to MS Teams. So how you get to the toolbar will vary depending on which version you're using. In the older version, you can make the toolbar appear by hovering your mouse over the meeting window. If you're using a newer version, the toolbar is permanently fixed near the top right of your meeting window. Regardless of the version, the icons should all look the same. They might, though, be in a different order than what is shown on my screen. A couple additional tips. If you're having trouble with your audio, check your device's settings, which you can get from the ellipses or those 3 dots on the toolbar. You can always use the meeting chat to contact the organizers, which is myself and my coworker, Amy Poles, and we'll do our best to help you out. And then when we get to it, the question and answer and discussion session, if you have a question, you have 2 paths to ask it. You can type your question into the chat and then I will read it aloud. Or you can click on the little hand icon on the toolbar to raise your hand, and then we'll call on you and you can unmute yourself and ask your question through the audio. All right, and now to kind of a little warm up to get us ready for this presentation and to use some of the tips I just gave you to use. We are going to do a quick interactive poll using Mentimeter. So if you'll all take a second to open up the chat, you'll find that my coworker Amy has posted a link to an icebreaker. Again, to get to the chat, click on the little speech bubble icon to open that chat sidebar, and you should see a link to the icebreaker question that Amy posted. If you'd humor me and take about 30 minutes to fill that poll out, I'd really appreciate it. And I will switch over to that so we can see those results coming in. All right, it looks like we've had a few people answer the question. If two heterozygotic black labs mated, what % of their offspring would have black, chocolate, or yellow coats? So go ahead and fill out your answers. I'll give you guys another 15, 20 seconds or so. We're just kind of figuring out how to use Teams, and again, it is, if you're having trouble locating the poll, it is in the chat bubble, and there's a link to it. Give you all just a few more seconds. We are getting some good results coming in. And I have the real answer for you guys, so don't fret. All right, it's slowing down. I'm going to switch back over to PowerPoint. So the real results, and you guys did a pretty good job, but about 56% of the heterozygotic black labs would have black coats, 19% would have chocolate coats, and 25% would have yellow coats. And so in the top right corner is a little picture of my dog, and it makes him a little more special because he's a chocolate lab. And now, Carl, if you will start sharing your screen, we will start our presentation. Great. Thank you, Carl. So Carl Ostberg is a research fishery biologist at Western Fisheries Research Center in Seattle. Carl's research focuses on the development and application of genetic tools in support of conservation and species recovery. His hobbies include skiing and vegetable gardening. Take it away, Carl.
Awesome. Okay, well, thanks. It's really great to be able to present here with this webinar series. Let me just, okay, so the goal of this talk then is to provide you with information about eDNA analysis for single species assessments, so you can get a better understanding about what goes on behind the scenes, all right? And so that's whether you're new to eDNA or someone who's not real familiar with the molecular and the analysis side of eDNA. So we'll start out with the talk with processes going from post-DNA extraction through data interpretation, and then we'll kind of shift gears and talk about uncertainty, and then identify some single species eDNA applications that are currently ready for use in resource management. Okay, so for single species detection then, we use species-specific markers to detect our species of interest. And these are targeted assays because they target a specific DNA region in a particular species. And it's important that we that these markers target DNA that occurs at high copy numbers in cells, because DNA is often in low abundance. So eDNA assays focus on mitochondrial DNA, high copy nuclear DNA, or chloroplast DNA, depending on the taxa. So you might ask, you can ask the question, then, so why use single species detection methods when there are multi-species methods that can provide information on communities in biodiversity? And that topic will be discussed next week by Taylor Wilcox in eDNA 301. But the answer to this question is going to depend on the research and monitoring question that you're addressing, how much sensitivity is needed for your study, what's the more cost-effective approach to accomplish your project goals. And so single species detection methods are they're ideal for cases where there's one or a few species of interest, and they can be highly sensitive and quantitative, And their most commonly applied eDNA method is shown in this graphic here. So this graphic shows the number of papers published using different eDNA methods and the year of publication up to 2018. So you can see that single species detection methods are taking the lion's share. But before we get any further, let's going to do a little bit of a review of some sample collection processes so that we can get to the point where we can begin our single species assessments. So we start by developing our sample design, and we identify what type of filters we want to use. We learned about that last week, and identify our sample locations and sample volumes and how many samples we want to take and all that. And then we go out to our sites and we collect water samples, and we're going to either filter on site or else we'll take them back to a lab or facility where we can filter. And when we're collecting water samples, we're also going to collect some negative control water samples, and these are distilled water, and they're treated just like a sample. And the negative field controls, they can tell us whether there may be some contamination during the filtration or preservation of the filters. And so then, we preserve the filters until the DNA can be extracted. And then DNA extraction is typically done using commercial kits, but there are other methods. And then during the DNA extraction process, we're going to include a DNA extraction negative control. And this DNA extraction negative control contains no DNA, and these controls can tell us whether there's contamination during the DNA extraction process. So what you're seeing here is that there's negative controls that are being incorporated into important stages of the process. So now we got our DNA extracted, we can test for the target species, and so we're going to do that by using PCR. But so let's talk about PCR first and kind of how it works, because I think we often kick around the term PCR without making sure everyone's up to speed on it. And PCR is integral to the eDNA analysis. So we'll start by looking here at conventional PCR, which is the general workhorse for PCR. So for the conventional PCR, we have two primers here, a forward and a reverse primer. And these primers are a string of nucleotides, typically 20 to 30 bases in length. And each primer is going to be complementary to a specific DNA sequence. So each primer sits down in a specific location and providing specificity to the assay. The chemistry also consists of DNA polymerase, which is an enzyme that synthesizes DNA strands complementary to the DNA template strand. And then there's free DNTPs, and there's buffers, and then there's cations also. So the PCR is going to have three stages. And this first stage is the denaturing stage, where double-stranded DNA is melted apart at high temperatures. So here we've got 95 degrees as a high temperature to melt apart the DNA. So at the second stage, during the second stage, the temperature is then reduced, and as temperatures cool, single-stranded DNA will reform or come together to reform double-stranded DNA. But what we're doing is we're reducing the temperature to an optimal point that allows the primers to anneal to the single-stranded DNA first. And so we're going to be doing this anneal at around somewhere between 54 to 64 typically. And so here the polymerases, the primer is going to bind, and then the polymerase is going to bind to the primer-bound DNA. And then there's a third stage, which is an extension, and this is where the polymerase synthesizes the new strand of DNA. And these stages are repeated, these three stages are repeated for 35 to 40 cycles, depending on your application. So specificity and efficiency of PCR is regulated by the chemistry and the cycling parameters, and in particular the annealing temperature. And so when PCR is performed under optimized conditions and amplification is efficient, amplification is going to, it'll happen exponentially. That is, there's a doubling of product every cycle until the reagents become consumed and amplification eventually is going to plateau. Again, this slide just represents conventional PCR, but I think it's a really good background to have to get us moving forward. Okay, so let's talk about some PCR methods that have been applied to eDNA now. So conventional PCR, just mentioned here, also referred to as endpoint PCR because the analysis occurs after PCR is run to completion. So typically, the amplified products are detected using agarose gel electrophoresis, which requires staining with a chemical to allow the DNA to be visualized. So that's kind of what's going on here is an agarose gel, and here's a band of DNA that's been stained. And this method was used early on in the days of eDNA species detection, but it's really not well suited for eDNA applications because it has poor precision. It's got low sensitivity, low resolution, requires post-PCR processing, as in like gels for detecting an amplification product. So it really doesn't get much use anymore. qPCR, or quantitative PCR, is the most common method, and qPCR is also referred to as real-time PCR because it allows the amplification of target DNA to be analyzed during the actual PCR process. Now, I'll talk about qPCR and the chemistry in greater detail shortly, but briefly here in this figure, the x-axis then represents increasing PCR cycles, and the y-axis here is representing increasing fluorescence. And so there's fluorescent chemistry is incorporated into the qPCR assay, and detection of a product, it occurs when fluorescence crosses above the background level, so kind of at about this level here in the graph. And so detection occurs during the run, and the benefits are qPCR are quantification of DNA, and that's going to be relative to standards that are incorporated in a run. There's high precision, high sensitivity. The qPCR machines are real common, and the methods are straightforward. And the third method here is droplet digital PCR, and this is a bit of a newer method. And so for this method, DNA and PCR chemistry, which includes fluorescent chemistry, similar to pretty much the same thing as qPCR, it's partitioned into about 200,000 individual droplets using an oil emulsifier. So here in this schematic here, you can see all these little droplets, and inside each one of these there's a PCR going on. And so each of these oil droplets then ideally contain 0, 1, or a few target molecules. And the PCR occurs in any of these droplets that contain DNA, and detection occurs at the end point of the PCR, where the fluorescence from each droplet is independently measured. So the number of positive droplets relates to the absolute number of starting molecules, and droplet digital PCR is able to quantify low concentrations of DNA with real high precision. And studies indicate that this method more accurately measures low concentration DNA than qPCR. However, these types of machines aren't very common, and so it is becoming more common here. So qPCR is the most common method that's being used, and so we're going to focus on that from here on out. So here, I just want to briefly mention samples, controls, and standards that would be included in a PCR run. So the graphic here is an example of a PCR plate, and what it's showing is all samples, controls, and standards are replicated on a PCR plate. So replication here is shown in triplicate. All right, there's and then samples are in black, and the samples are replicated because the eDNA often occurs at low concentrations. So it helps you to better estimate the eDNA with and detect it when you're replicating it. And then there's negative controls. You know, we're including our negative controls that we've been accumulating throughout the process. We have the field negative control. We've got the DNA extraction negative control. There's also a no DNA template control, and that is it's just DNA that, or sorry, it's just a sample that has water in place of DNA. And we also have our standards here, and these are serial dilutions that allow you to estimate the DNA concentration in your samples. Standards are going to also let you know if your PCR is working and how efficiently it's working. So the example here, we've got 5 10-fold standard dilutions that are starting at 100,000 copies and going down to 10 copies. And so replication of the standards just produces a much tighter standard curve. So yeah, so let's talk about the single species detection assays we typically use with qPCR. And this is the hydrolysis assay, or also called PNAMP assay. And this is what we're using, typically using for single species detection with qPCR, and also with droplet digital PCR. So these assays are typically 80 to maybe 200 base pair in length. And the chemistry is similar to the conventional PCR, with the exception that we have a probe that's being added here. Okay, and the probe consists of a string of nucleotides, about 18 to 25 bases in length. And like the primers, the probe is complementary to a target DNA sequence. So it's going to bind to target DNA. And so the probe also, it provides an additional layer of specificity to the assay, right? So the probe has a fluorophore that's attached to the 5' end, which acts as a reporter, all right, at this point. And then there's a quencher on the 3' end, which quenches the fluorescence when it's in close proximity to the reporter. And there's also polymerase and DNTPs and buffers and in cations in the mix, and they come as a master mix. And so there may be many different master mix varieties that have different attributes, and they're available from different vendors. So then during the PCR, qPCR assay, there's, you know, we're cycling also, and we got these temperature gradients, and DNA is being denatured, all right, so, being melted apart here at the high temperature, and then you know, as the temperatures reduce, we get into primers that are binding to the DNA, and the probe is also binding. And what you'll notice here, you can see that the probe is sitting down between the it's strategically located to sit down between the forward and the reverse primers. And so the polymerase then begins to synthesize a new DNA template strand during the extension stage. And the polymerase also has an exonuclease activity that degrades the probe when the probe is bound to the DNA. So probe degradation, the degradation of the probe allows for the separation of the reporter molecule from the quenching effects, and that reporter to fluoresce. Okay, so now let's take a look here at the at a qPCR amplification plot profile across cycles. So understanding this profile will help us to make sense of the qPCR output. And so this figure shows 4 major phases of qPCR amplification. And on the x-axis is the number of cycles, and it's going from like 1 cycle up to 40 cycles. And the y-axis is a measure of the fluorescent signal. And so it begins with this linear ground phase where the PCR is just beginning. Fluorescence from the PCR hasn't risen above any kind of background detectable level yet. And then we start getting increase in product, and we enter an exponential phase. And this is with the products being doubled every cycle, and there's an exponential increase in the fluorescence. And this is a real important phase, and we want to have high efficiency in PCR to ensure doubling of the product. Then we go into the linear phase, and that occurs as the reagents begin to be consumed and the reaction slows, and then we get into the plateau phase up top as reagents become exhausted. Here, now we're looking at real data from the qPCR output. And this graph shows a qPCR amplification plot after a run. And like the previous slide, the x-axis is the cycle number, and it's going from 1 up to about 45. And the y-axis is a measure of the fluorescent signal. So you can see there's this green horizontal line that's going across the graph. And this is the point where fluorescence that's being generated by the PCR product is being reliably detected above the background noise fluorescence. So there's going to be this background noise fluorescence going on, and in order to differentiate background from true product, we set a threshold. This is the point in which so the point here is where the sample is going to be when it crosses this threshold, it's going to be referred to as the quantification cycle, or as the CQ, or the cycle threshold, which is CT. So detection of a product in a sample occurs at the cycle where the accumulated fluorescence then is going to be exceeding the background level. So the graph here shows 5 serial dilutions of a standard with having 10 copies up to 100,000 copies. And so the more concentrated DNA then is going to reach a CQ a lot earlier than less concentrated DNA. All right. So this means that less DNA, so that when there's less DNA, the CQ is higher. So basically, detection and non-detection results can be inferred based on whether fluorescence of a sample has crossed the CQ. Now, a standard curve can be generated using the CQ values from the DNA standards. And so for the standards here, CQ values are on the y-axis, and the concentrations are on the x-axis, right? And then we can estimate the DNA copy numbers in our samples by using a regression equation that we develop off of these standards. So we've got 2 ways to interpret qPCR results: the detection and non-detection of a target species sample is going to be based on whether the fluorescence in that sample crosses a fluorescence threshold, and then we can quantify the target DNA in a sample based on the standard curve. Okay, so so let's look at detection and non-detection interpretation in a bit more detail. So eDNA is often found at low concentrations in environmental samples, and detection is reduced at low samples. For example, this figure here shows the amplification of 3 dilutions of DNA, 1, 10, and 1,000 copies for 8 different assays here, and all these 8 assays are in different colors. So for each assay, there are, say, there's at least 20 replicates here at each concentration. So the y-axis is going to be the frequency of positive PCRs, and what we're seeing here is that at these low concentrations, say 1 copy, no assays produce 100% positives. All right, and this is because at low concentrations, you might not always be pipetting DNA into your PCR, although you supposedly have, you should be able to get 1 copy, but you're not always pipetting 1 copy. So as the concentration increases, there's a greater chance of pipetting DNA into the PCR. So then at 10 copies, most assays are amplifying at 100%, just about, and at 100 copies, all assays are amplifying at 100%. So variability in pipetting low concentrations of eDNA means that detection is going to be affected at low concentrations. So So what does reduced detection at low concentration mean to an eDNA sample? So if we run triplicate PCRs on a sample, there are going to be four possible outcomes, right? So you could get all three replicates could be positive, as in this first sample, or all three replicates could be negative, as in the second sample. And these two are easily interpretable as detection and non-detection. The two other outcomes are imperfect detections, where only one or two of the replicate PCRs are positive. And these imperfect detections, it might create some uncertainty as to whether the original water sample should be scored as a positive or negative. There really is no standardized method for interpreting and scoring imperfect detections, and the stringency of scoring positive results might depend on the sample design and on the research and monitoring question. So now let's look at interpreting DNA concentration results in more detail. So ultimately, we'd like to estimate DNA concentrations in our eDNA samples. But first, we're going to need some information on assay performance at low concentrations, which can be described by the limit of detection and limit of quantification. So the limit of detection, that's going to be the lowest standard concentration in which 95% of replicates are reliably detected. And the limit of quantification is the lowest standard concentration that can be reliably quantified with a defined precision, say like maybe 35% coefficient of variation. And the LOD and the LOQ for an assay are estimated using a dilution series, like it's shown here in this figure, in this plot here. And so this plot is a standard curve from an eDNA assay, and it's showing the standard concentrations and their CQ values. And for this example, we have 40 replicates at each concentration, say. So the LOD and the LOQ can be estimated directly from these standard concentrations that we have, or we could estimate them using a curve fitting method, which isn't limited to only the standards. And there's a great paper by Klimas et al. That provides a really nice curve fitting method, and it's really a great paper. So here, like say in our example, let's say we use a curve fitting method, and the LOD is estimated at 8 copies. So that's where 95% of the replicates are going to be detected at this concentration. And then the LOQ with a 35% coefficient of variation as our defined precision is going to be estimated at 40 copies. Then eDNA concentrations that fall above the LOQ are reliably quantified, and they are going to be acceptable as quantitative data. And the idea is that we'll have a reliable estimate of the number of eDNA copies for samples that fall above the LOQ. DNA concentrations that fall below the LOQ then may be considered as unreliable because there's too much variation in the DNA concentration among the replicates, but the results are totally acceptable as qualitative data. So that would be like a detection, non-detection, or plus minus, or whatever you want to call them. And so the LOD, LOQ need to be determined for every target species, and they may vary between labs. So, you know, they should be figured out at each lab that's using these markers. So the last thing that then I'm going to talk about that's specific to PCR inhibition. Sorry, the last thing I'm going to talk about is specific to PCR is PCR inhibition, and it's not uncommon for PCRs to be inhibited by compounds that naturally occur in the environment, like humic acid or tannic acid. And so the first thing we do prior to testing our target species is to test for PCR inhibition. And testing is performed using an internal positive control. So this is done using commercial kits, or samples can be spiked with control DNA that's not found in the sample, or you don't expect to see it in that in those samples. And so an uninhibited internal positive control is expected to come up at a specific and consistent CQ. Okay, so in the figure here, this black curve is a PCR that's uninhibited. Okay, so this dashed line then represents the fluorescence threshold, and inhibited PCRs are going to cross this fluorescence threshold at higher CQs, or they may not cross at all. Okay, and so all of these samples here are going to be inhibited. And so, but inhibition can be overcome by using commercial kits that clean up DNA, or by diluting your sample DNA once that those inhibited samples are identified. And there are some of the qPCR master mixes are more resistant to inhibition effects, so you may see differences depending on kind of what kind of master mixes you're using. Okay, let's see. So at this point, maybe you've recognized that there's some uncertainty and potential errors in the DNA analysis, and I think it's captured really well here in this quote, and this is from a paper by Jardy from 2019, and a really a good paper. And it's an anonymous fisheries manager tasked with preventing Asian carp, spread into the Great Lakes, told me, "I will never believe in eDNA positive detection until we capture a fish." Now, I don't know how recent this quote is. The paper's from 2019. I mean, it could be a quote from 2018 or 19 or maybe 2012. But, you know, I think it's not an uncommon viewpoint. And I think that eDNA practitioners need to communicate and be transparent about sources of error and how we try to mitigate it. So at this point, I'd like to dig into some of these sources of uncertainty for single species assessments. So uncertainty is inherent to the eDNA analysis, and uncertainty resides in potential for errors in detection and non-detection. And potential errors could result in false positives, so in the species absent and DNA is detected, or result in false negatives where the species is present but DNA is not detected. And, you know, but this uncertainty and error isn't just specific to eDNA methods. I mean, any sort of sampling method is going to have some errors in it. But anyhow, let's decide. I kind of want to dissect out these errors and the uncertainty for the eDNA analysis, and how we might, kind of what their causes are and how we might mitigate against them. So contamination, yeah, that's probably the biggest contributor to false positives. And so this can occur during field sampling, filtration, or any point in the lab analysis. So contamination can be minimized by following best practices and ensuring proper quality assurance and quality control. So this might include, this includes thoroughly cleaning and disinfecting sampling equipment, lab equipment and benchtops, and having specific lab spaces, hoods, or rooms that are dedicated to each of the sample preparation procedures. And then, you know, also having equipment that's dedicated to each of these lab spaces, that they don't move from the lab space where their home is. False positives could occur if eDNA assays are detecting non-target species. And, you know, and that would most likely be a species that's closely related to the target species. And so this could be mitigated by rigorous testing of the assays against non-target species that might co-occur with the target species, and which is done using DNA sequence information in GenBank and testing against tissue samples also. But this is typically done when species assays are being developed and validated. There's allochthonous DNA, and that's eDNA that's transported from a source outside the sample area. So that could happen, say, through feces from a predator or a waterfowl, maybe a bird, waterfowl's flying from one body of water to another, or maybe boats. Someone's boat could be transferring DNA or cleaning a fish, or a lot of different ways. But I mean, this is a source that you really can't mitigate against. And then lastly, eDNA from a target species could persist in the environment, although the target species isn't actually present in the environment at the time of the sampling. And so this could happen if, say, if the target species recently passed through the area, or if eDNA that's settled into sediments becomes resuspended into the water column after some events, flooding or storms that might stir up sediments. So this is why it's important to avoid stirring up the sediments when you're sampling eDNA, and if your target species is passing through your sample area, it's just kind of being transient. Maybe temporal sampling could be helpful for this also. So for false negatives, then, PCR inhibitors could cause false negatives if they're not accounted for. And we just kind of talked about that, but like I mentioned, it's common protocol to first test for PCR inhibition prior to testing for the target species. So inhibited samples are going to typically be identifiable. It's possible that your assay could fail to amplify in the target species, and this could happen if there's genetic variation, say, in the target species, so that the primer and the probe don't completely match the target sites. And so sometimes assays are developed with limited geographic coverage. It's a good idea to have an understanding of how the assay was developed and the geographic extent of the tissues that were used to validate an assay. You might need to test the assay on tissues that are obtained in your sampling area if this wasn't done. I mean, it's always a good idea to, when using new assays, to be testing those assays on tissues on the target species and non-targets, just to make sure. And then eDNA could be in false positive, could result from eDNA in low abundance. So recall that eDNA can be difficult to detect when in low abundance. And so this could happen when species are rare or occur in low numbers, or maybe samples are taken too far away. So detection is going to be improved by performing replicate PCRs. And that's why we do this replication. And detection can also be improved by increasing the number of sample sites, number of water samples collected, or increasing sample volume. This is a good point to mention some strategies for managing uncertainty. It's a good idea to have detection criteria established prior to collecting or running samples on qPCR. Repeatability of results could be assessed by rerunning DNA samples, resampling water, or by including spatial or temporal sampling in the study design. Applying a non-molecular sampling technique with a detection method that's comparable to eDNA could be used to verify questionable results if that's possible. There's occupancy models that can be applied to account for imperfect detections. You could maybe model probability of false positives and false negatives in your sample collection. And I think decision trees can be really useful if there's an expected management outcome as a result from coming out of a eDNA survey. And I think the take-home message here on this slide is that it can be helpful to have a plan prepared for managing managing uncertainty in the eDNA analysis. Okay, so I'm going to shift gears here a little bit now. So single species eDNA assessments have great application to resource management. And it's been established that single species eDNA assessments are robust at detecting target species when compared to traditional sampling methods. And the speed and the ease that eDNA surveys can be accomplished relative to traditional sampling methods is a major attraction of eDNA analysis. So here I've listed some major eDNA applications that are relevant to resource management that you could pick up and run with today. And so I'd like to talk about how eDNA is being used with these applications. So the first one is early detection and of invasive species. So invasive Asian carp, like start off with a invasive Asian carp as an example. So they become widespread throughout the Midwest, and since about 2010, large-scale eDNA surveillance has been part of the Asian carp monitoring program here, and it's aimed at preventing the spread of Asian carp into the Great Lakes region, or into the Great Lakes themselves. And since the beginning of the Asian carp eDNA surveillance, there's been a quality assurance plan in place that gets updated periodically. And the plan serves as a guide that standardizes field and lab methods that are employed during the Asian carp monitoring program. And so this plan then, it requires that before a site can be determined as being positive, a general assay, qPCR assay, is performed that targets both bighead and silver carp. And at least one replicate eDNA needs to be positive. Okay, and so we start here with this first. We're starting off with the general assay here, and then if there is a positive that's in that assay, then that triggers a second assay to be performed, and that is an assay that is going to be specific to the bighead carp, and then another assay that's specific to the silver carp. And this will require one positive PCR as being positive. And so each of these assays that are targeting different parts of the mitochondrial genome. So here there's a redundancy is being built in to remove any uncertainty in this eDNA surveillance program for Asian carp. So closer to home here in the Pacific Northwest, there's a lot of invasive species that are of concern. Some established, others have yet to invade. And if they were to invade and become established, it could cause some real significant biological and economic impacts. So take, for example, the zebra and quagga mussels. So while they're not in the Columbia River Basin and not in several western waters, there's recognition that the zebra and quagga mussels could cause extensive damage and burden if they were to become introduced. So recent detections then in Montana waters and interception of infested boats highlight the need for measures to prevent and contain the spread. And this is recognized in the document Safeguarding the West. And in this document, eDNA is recognized as one of the actions for early detection monitoring. And so the current standard then for early detection of zebra and quaggas is to capture the veliger larvae by using plankton nets and then microscopy, and then confirm species with eDNA tests. The application of this method is limited to the time after adults are spawning and while the larvae are still in the water column. And the eDNA analysis has two advantages over the standard method, and that's that there's fewer methods involved, and then there's a wider temporal sampling window, so that several studies have shown that the zebra and quagga eDNA can be sampled and detected outside of the time when the larvae would be expected to be in the water column. And so eDNA sampling, say in the fall or in the winter, could allow areas to be prioritized for non-molecular methods in the spring and fall. So single species assessments are commonly used to map species distributions, providing some results that are significant and important for conservation efforts. And I think here's a nice example is identifying eDNA distributions of northern pike in the Columbia Basin, and that's here on the the right-hand side of this slide here. And this figure then here represents the observed distribution of northern pike based on angler and gillnet surveys. And so the red circles, those indicate sites where northern pike are present. The orange triangles are sites where the northern pike could possibly be present, but they're not sure, but possibly based on proximity to the sites where they're present. And then these white squares are sites where northern pike have not been observed through angling and gill netting studies. But what's interesting here is that so eDNA was sampled in these areas, same areas, and so what they found was that it was detected at all the sites where the northern pike were known to be present, but it wasn't detected at any of the other sites. So, I mean, this is really cool, I think. So for cases of non-native species, and like, say, for example, those species that are really voracious, like a northern pike, eDNA distribution maps could be used to inform resource managers where to prioritize suppression efforts, if that's a management strategy. eDNA has great application for monitoring change in species distribution over time, changes due to a management action like barrier removal or habitat improvements or eradication efforts. So here, just want to real briefly talk about a monitoring study that tracked recolonization in the Elwha River following the removal of 2 dams. So the 2 dams on the Elwha River, they blocked fish passage, were recently removed, and a key objective, a management objective of this removal was that there was fish passage above the former dam sites and restoration of connectivity between the lower and the upper river. So in effect, success of the dam removal is going to be judged by salmon recolonization. So eDNA was monitored to infer the spatial extent of Pacific salmon, lamprey, char, and trout following the removal of the 2 dams. And samples were collected from 2014, 2017, over several months and per year, and depending on accessibility to the sample sites. And so summary results for Chinook salmon DNA are shown here on the right-hand side. And they indicate that after the removal of the dams, Chinook salmon were accessing habitat far above the former dams. So you got the 2 dams here, and right here and here. So here in this light blue outline that represents the historical Chinook salmon distribution. The orange outline represents their distribution prior to the removal of the 2 dams, so they were limited to about the lower 8 kilometers of the Elwha River. And then the yellow outline represents the distribution of Chinook salmon after it's the Chinook salmon eDNA after dam removal. So it's pretty neat. And then so occupancy modeling was also applied to this eDNA data to estimate the probability of occurrence of eDNA at a site. And so the models then, they were applied using data collected at the time when adult Chinook and coho salmon were returned to spawn for each of the sampling years. And those sampling years, you can see them at the top here. So the x-axis then is the distance from the river mouth, and the y-axis is the probability of eDNA occurrence. And so the results indicated that the probability of detecting eDNA of Chinook and coho increased upstream over time, suggesting that adults had accessed habitats further upstream in higher numbers from the beginning to the end of the study. And so from a management perspective, it seems that eDNA data indicate that the objectives for dam removal are being met. There is fish passage and connectivity. So biosurveillance of pathogens has great application. And so it's got application to natural environments where viruses, bacteria, and parasites are being surveyed in aquatic systems. And these types of studies are investigating how environmental factors are driving pathogen loads in natural systems. And biosurveillance of pathogens is highly relevant to salmon and steelhead hatcheries in the Pacific Northwest. So within hatcheries, it can be used as an early detection tool, assisting in the rapid response to disease outbreaks. And if an outbreak is caught early on, management actions could minimize mortality, and there may be less need to put fish on antibiotic feed, which could reduce selection for antibiotic-resistant bacteria in the environment. And so I talked a little bit about occupancy modeling, but now I'd like to talk about it in a little bit more detail. And so occupancy modeling can be applied to most eDNA applications, given a proper experimental design. And so eDNA occupancy models consist of 3 nested hierarchical levels, and that are depicted here in the figure on the right-hand side. So this first level then is sample locations, and you can see the sample locations are on the Oregon and California coast. And so the parameter that's estimated here for the sample location is the probability of eDNA occurrence at a sample location. And you can think of that as occupancy of eDNA. And then the second level is water samples that are collected at each sample location. And so here you've got this, got 6 water samples here. Each of these little circles represents a water sample. So there's 6 water samples at this one specific location. And so the parameter here that's being estimated is the conditional probability that eDNA is collected in a water sample, given that eDNA occurs at the sample location. And you can think of that as being the availability of eDNA in the water. And then there's a third level, and that's PCR replicates in each water sample. And so here you've got each of these water samples. There are 3 PCR replicates represented. They're kind of slices of the pie. And in this water, this location, you've got detection is a gray, filled in gray, and white is non-detection. And so the parameter that's being estimated here is the conditional probability that eDNA is detected by qPCR, given that eDNA is collected in the water sample. And so you can think of that as being detectability of eDNA. So covariates can be applied to the occupancy model to determine their effect on eDNA. And the figure here on the left, it shows the probability of Gobi eDNA occurrence in a water sample on the y-axis, and salinity on the x-axis. And what we see, that the probability of the Gobi DNA in the water samples is decreasing with increasing salinity. And models can be used to assess the probability of obtaining and detecting eDNA. So the figure on the right shows how the probability of collecting eDNA at a location increases with the number of water samples. So there are 4 water samples here, and the results show that your probability of collecting eDNA using the same number of water samples might differ among sites. Okay, so kind of wrapping this up then, I'll wrap this up by talking about single species methods and applications that might be a bit on the horizon or that we can look forward to. So I haven't talked about using eDNA to estimate abundance and biomass. And I mean, there's a great interest in this application with resource managers. There's active research on this topic, several papers that demonstrate significant relationships between eDNA concentrations and population abundance. However, the current state of this application is not at the level of any of these other applications that I've been talking about. And there's a lot more research and refinement is needed to get to this point. Now, I know there's a lot of resource managers, they really want to know, can we use eDNA to, can you tell me how many fish I have if you do an eDNA analysis? But, we're really not there yet. Coupling eDNA to USGS stream gauge networks, a couple of really nice papers have called attention to coupling eDNA to stream gauge networks, and that seems like a really good next step in the monitoring process, and particularly if automated eDNA sampling was incorporated into the stream gauge network. And portable automated samplers could be moved around to strategic locations depending on the project. And rapid on-site detection currently exists, but it'll likely become more common as the need for results in real time increases, or as field biologists become more involved with testing of samples. And then lastly, research on environmental RNA is really starting to ramp up, and eRNA degrades more quickly than DNA. So eRNA might provide a better measure of real-time presence than eDNA. Since RNA is a transcribed gene product, eRNA could provide information on the physiological and health status of target species. All right, so with that concludes my presentation, and I'll be happy to take any questions if there are any, or discuss.
Thanks, Carl. And a quick reminder, if you want to ask questions, you can type them into the chat and I will read them aloud, or you could raise your little hand on the toolbar, and then we'll call on you and you can unmute yourself and ask them. All right, it looks like we have a question from Steve. How long does eDNA from a migratory fish persist in, say, an estuarine bay environment?
Well, it's a good question. It's a really good question, and I'm going to say that it kind of depends. And, it'll depend on the tides, what the water flow is like, how much water is moving around. Are those what the kind of currents are moving that away, or the eDNA move, how that's moving through the system or through that estuary, what kind of breakdown, enzymatic, bacterial, or breakdown, what kind of processes are breaking down the eDNA. I don't know offhand whether to say, how many days or hours, but it's going to vary by the environment. And it's certainly being detected out in salt water. There's studies where they're detecting eDNA, so it's persisting, but for how long, I don't know, whether days or how long. So, but it's going to be dependent on a lot of factors.
Great, thanks. We have another question coming in. How far can intact eDNA be transported and detected downstream in flowing water systems? Wow,
yeah, good question. Again, that's going to depend a bit. The studies have, there's been a lot of variation in studies as to what they've shown. One study, I think it was as high as like 6-7 kilometers, others much less. It depends on the species, the density of the species, depends on water flow. It depends on if it's a slow moving water, maybe it'll be settling out, the eDNA will be settling out a lot more than if it's kind of a faster moving water. Depends on water temperatures, and these are good questions, and these are the types of questions that a lot of folks are really actively working to try to understand and address. But yeah, so I mean, those are really good questions, and I think there's some studies that are suggesting maybe you should be sampling within a kilometer or half a kilometer distance and things like that. But I mean, there's a lot of these answers are just a pain.
Great. We got another question from Roy Morris. What might we learn from RNA that we won't learn from DNA? Yeah,
so RNA is great. RNA is so DNA is transcribed to RNA. So what that means is that it's on its way to becoming some sort of a product, whether a protein or a hormone, or it's going to be some sort of a biochemical and a biological molecule that's going to have some sort of significance to that animal. It could be an immune response, maybe, that it can tell you about whether the fish is spawning, but so the RNA is it's a direct link to a function that's happening. And the DNA is just the code. It's like a book, but that RNA is a specific product that's coming out of that book, and it tells you. It can tell you about the physiological status of a fish. It can tell you what the health is, whether they're fighting a disease. It can tell you whether they're maybe stressed, if there's like hot water in the Columbia River, if they are really being stressed out. There may be high expression of heat shock genes, things like that. So I mean, it tells you a bit more about the biology of the organism than just straight eDNA. But I think that eRNA is going to be more. It'll have application to single species, or these single marker species methods, but it'll have more application, I think, with kind of more of a sequencing approaches, which maybe Taylor might be talking about next week.
Very cool, thanks. We have a question for Marlies, or from Marlies. What are the most common mistakes by those that might be familiar with molecular genetic studies, but are eDNA novices and plan their first eDNA study?
Yeah, I think cleanliness, maybe. Just doing everything on one benchtop, all of your methods on one benchtop. I think it's really important to have separate spaces for eDNA, where you're doing the DNA extraction at one place, you're pipetting out standards in another, you're pipetting out your master mixes in another place. Just having separate rooms, that can be really good for removing any contamination. I think contamination is going to be kind of one of those big things. Yeah, I think that would be really important, is kind of separation of space and equipment. Yeah, that's an interesting question, but yeah.
Perfect. Yeah, we have another question from Roy Morris, and it's a little specific, but maybe you can help. Where would the work be done to differentiate Hoh River steelhead from Keats River steelhead?
Yeah, so that is, right now, that's beyond kind of the eDNA application. Right now, it would be really cool to get there, and hopefully we can get there. But that's a real, that's a population genetics question there. And so, when we're figuring out the population genetics parts, we're taking tissue samples from discrete individuals, and we're isolating that DNA from each individual into a separate tube, and that's being tested. Everything's being tested separately, and so we can keep individuals separate, and so we know what their genetics are, and then when we look at their genetics from one population as a whole compared to the genetics of another population as a whole, then we can start looking at these differences between populations. But the eDNA itself is much more challenging because we're sampling a big pool of individuals, okay? And so eDNA is going to be targeting mitochondrial DNA. And so right now we're just targeting one single marker that's going to be, we want it to be identical between Queets and whole river fish. We want that to be identical, all right, when we're using eDNA. But so we're also, while we're out collecting that eDNA, we're also collecting nuclear DNA, and that's the nuclear DNA is what you're looking at when you're looking at the population genetics of between these two populations. But these water samples that we're collecting are a huge. It's you know, you collect one water sample and it's a soup of every individual that that you're detecting in that environment. So in the proximity of your sampling. And so I mean, you can have one individual or you can have 100 individuals contributing to that soup of eDNA or more. And so until you can isolate each of those individuals from that water sample, I think you can have it's going to be very difficult to do any population genetics. Made sense.
Great, yeah. We have another question. Are there studies of eDNA in eRNA comparing hatchery and wild fish?
You know, honestly, not that I'm aware of. Maybe somebody else knows, but I can't. Not that I can think of. I think the eRNA is just in its infancy right now. I mean, it's just starting to get, people are just starting to look at it. So it's pretty out there right now.
Yeah, and our next week's presenter, Taylor Wilcox, just chimed in. He said very few studies of eRNA at all right now. So just to reiterate what Carl said. We have time for a couple more questions if they come in. Looks like they're slowing down. So I just want to say thanks to everyone for attending and for all of the great questions. And a huge thanks to Carl Ostberg, our presenter. We did record today's webinar, and the video will be posted to PNAMP's YouTube channel in the next couple of days. And there'll also be a link on the environmental or the emerging technologies information sessions landing page on pnamp.org. Next week, we'll continue the eDNA webinar series with a presentation by Taylor Wilcox, and I'll show that schedule here in a second. The schedule for future webinar series will be in upcoming newsletters. We release these newsletters usually at the beginning of the month. You might have just got one in November. If you don't get the monthly PNAMP newsletter, Amy, my coworker, has put a link in the chat for the sign-up form, so you can join there. And then additionally, we post all updates to the schedule on the ETIS landing page as they become available. Also, if you haven't already, consider joining the eDNA community of practice, where you can make connections, share information, and start collaborating on work related to eDNA methods, tools, data handling, and resources. And Damian Manning, he has put a link to that also in the chat. And there's a link on this screen too, which you cannot click on, but it's in the chat. Thanks, Damian. And you don't have to be a DOI or USGS employee to to be part of the community of practice, which is great. So here's a schedule for the rest of our eDNA webinar series. Last week, we had David Pilliard and Matthew Laramie, and that recording is already up on YouTube and on PNAMP.org. Carl's presentation will be up there in a couple of days. And next week, we'll have Taylor Wilcox from the National Genomics Center for Wildlife and Fish Conservation. And he'll be presenting our eDNA 301 webinar, Multi-Species and Biodiversity Assessments, focusing on laboratory procedures and interpretation of results, including challenges and future directions. The format will be similar to today's. It'll be approximately 45-minute long presentation followed by question and answer, and it will start at 1 Pacific and go to about 2:30. And that concludes today's webinar. Thanks for sticking around. We hope you join us next week.